Alright, ladies and gents, welcome back. We're going to talk about our final topic as it relates to water chemistry, and that's this idea of pH and acids and bases. So here's the interesting thing. When we look at water in and of itself, in its purest form, if I were to stick a beaker in front of you and strictly have 100% H2O in that beaker, you've got this interesting phenomenon that happens with those water molecules as they're floating around in that beaker. And that's the fact that in any sort of aqueous solution, whether it's just the water itself or we start mixing in some sort of solvent, you're going to get this small percentage of water molecules that dissociate or break themselves apart. And that's what this chemical reaction is showing down here. So here are two water molecules, right? Here's your hydrogen bond, maybe not a bad thing to label in your notes for yourself. And what ends up happening is that that hydrogen bond every now and again tugs just hard enough and yanks off this hydrogen that gets then attached to this particular water molecule. And what we end up creating are two ions in solution. Your hydronium ion, which is an H3O+, right? I've got that extra proton, that nucleus from that hydrogen, but in the process of yanking this, it takes the hydrogen nucleus with it, but leaves that extra electron around that oxygen, and so you get a hydronium or H3O plus ion plus a hydroxide ion or an OH. And when we talk about pH, they often shorthand this, so you don't have to write H3O plus every time, and they just talk about it as a hydrogen ion or an H plus. And really this is a reversible reaction, so we should add another arrow to this, because just as this is happening where that dissociation is occurring, and you're getting those H plus and OH minus ions, they're also reconnecting to come back to this direction and make water. So adding that extra arrow on here would be a better way to really indicate what's going on in solution, because they're constantly coming apart and remaking themselves. So how does this relate then to acids and bases? Well, that comes in to play then when we start talking about the actual solution process, so water being that solvent, but then what else am I dissolving in that solvent? So an acid really is a solute that we're dissolving in water, and as a result, when that acidic compound dissolves in water, it itself comes apart, dissociates, and ends up adding extra H pluses to the solution. That's what makes it acidic. Something like hydrochloric acid, so HCl comes apart in water, and now I've got these extra H pluses in solution from the hydrochloric acid and those chloride ions. Alright, so that adds extra H plus to solution. And we get a certain set of properties that is often associated with acidic compounds. Things like a sour taste. For any of you who like the Warhead candies, take a look at the actual back of the candy package, and what you're going to see there more than likely is that they use acetic acid, or citric acid I should say, and they just pump and coat those candies with tons and tons of citric acid. Naturally we find that citric acid in a lot of your citrus fruits like oranges, alright, and they've just concentrated it into powder form, which is what kind of sucks your cheeks in, gives it that sour taste. Vinegar is another common acidic compound, specifically acetic acid. Alright, now what you're typically eating on your french fries is a very watered down, diluted version of acetic acid, but once again, where do you get that sour taste from? It's because it's acidic. They're also known to be very corrosive on metals, so they'll sort of break them down at very concentrated forms. So, acids, sour taste, corrosive on metals, and they're acidic in the sense that they are adding extra H pluses to solution. The opposite then are your bases. And basic compounds are once again solutes that I'm dissolving in water, okay, making that solution, but now these bases are going to remove H pluses from solution. And they can do that actually in one of two different ways. Either they're going to remove those H pluses because there's something like ammonia, alright, and ammonia is NH3, but it's actually more stable as an ammonium ion, which is NH4+. And so they suck those H pluses out of solution, they suck them off of water molecules, making this NH4+, and because I now have less of those H pluses in solution, I can't recreate the water molecules, and so I end up hanging out with excess OH minus. The other way that we can do this is by having the compound itself disassociate and leave extra OH minus in solution, which can also suck H pluses from water to make more water. Something like sodium hydroxide or NaOH, that's what's going on here. It's an NH plus and an OH minus. Now I'm going to increase this, which means it can now connect with some of those H pluses from water, making more water. Alright, but you get this disproportionate value of H plus to OH minus that you would typically see if it was just water. Okay, so they're going to remove H plus from solution as opposed to the acids that add them. So what sort of properties do we end up seeing with bases? Well, unlike the acidic, sour taste we get with acids, we get this bitter taste that gets associated with bases. If you've ever eaten unsweetened baking chocolate, it's incredibly bitter. It's not like eating a Hershey's bar. And that's because those cocoa beans contain a particular base that gives them that bitter taste. Alright, and we mix it with milk and sugar and do all that kind of stuff to make the Hershey bar and make it palatable to you. Another example is that most bases have these slippery feels. A lot of cleaning products, soaps, Lysol, Clorox, that kind of stuff, they've all got this slippery feel to them because they're all containing bases. Okay, so how do we end up measuring an acid versus a base? Well, a lot of times we'll use indicators. And an indicator is any sort of substance that can change color in the presence of a base, and we've got a whole host of indicators that we might use in various situations. If you've ever used the little blue or red test strips, alright, that's litmus paper. The blue litmus paper turns red in the presence of an acid. If it's in the presence of a base, it'll stay blue. The red litmus paper, on the other hand, will change blue in the presence of a base while staying red in the presence of an acid. Litmus paper is great if all you want to know is whether it's acidic or basic. Now, we've got pH strips that we use a lot more probably at the high school level. You may use that litmus paper more at a middle school or elementary level. The pH paper then actually gives you a range of colors based on how acidic or how basic you're dealing with. Another common one that you may come across in chemistry class is this liquid called phenolphthalein, and if you ever do titration experiments in chemistry, you'll come across this one because that's a common place for this to get utilized. Colorless in an acidic solution, alright, but you can actually titrate or do drop by drop by drop to figure out at what point do I change my acidic solution over to a basic one. And you talk about them, those titrations are often trying to find what we call a neutralization point. So how much of a particular indicator do I need to add is going to indicate how strong of an acid I'm dealing with, right? So it'll stay colorless until you've reached that neutralization point, or just cross it, and that's where it becomes then red in a basic solution. And we've got natural indicators. If any of you have these hydrangea flowers at home, we can tell the acidity of your soil based on the color that those hydrangeas then grow in that soil, assuming, of course, you're not doing anything additive to the soil itself. So if you grow hydrangeas in acidic soil, they'll end up producing these bluish-purple flowers. However, if they're grown in a basic soil, which is a lot of what we have around here with the clays and things like that, alright, they end up being pink. So all my hydrangeas at home happen to be pink because there's lots of clay in my soil at home, and so that tells me that my soil is very basic. Now, if I wanted my hydrangeas to be this color, I would have to add some acidic compounds to the soil to make them that color. They wouldn't naturally grow that way without some help. And if you see a combination really of blue and pink flowers on the single plant, that tells you that your solution really isn't acidic or basic. It's kind of in the middle, something we talk about as being neutral. So solutions are defined based on acidity or basicity, really on the concentration of those hydronium H3O+, or if we simplify it to the hydrogen H+ ions. And as a result, we can then also tell what the hydroxide ions are, or your OH minuses. So acidic or basic directly connected to this guy here, your hydrogen ions, and then we can infer those hydroxide ions. Maybe not a bad side note for yourself there as well. So if I take a look at pure water, I know that if it's 100% H2O, the only possibility is to have an equal concentration of H+ to OH-. Split the water molecule apart. That's all you can create. So we talk about that as being our neutral point. Okay, so it's not acidic, it's not basic, it's neutral. Once I start adding things to that water then, I can see how those relationships between H+ and OH- change. Now keep in mind that they're directly proportional to each other. So if I put something into that solution that causes my OH- concentration to go up, then my H+ concentration has to do the opposite and go down. Take note, by the way, that these little brackets here, alright, indicate concentration or how much of that ion exists. It's just a shorthand, so I don't have to say the concentration of OH- all the time. Alright, it's just a chemistry shorthand. So what we end up doing then is looking at pH in terms of the pH scale, right? So here's 7. Here's your pure water. Alright, any substance that exists in which that H+ and OH- concentration are equal to each other, like water, although it doesn't always have to be water, are neutral substances. So they are not acidic, they're not basic, they've got an H+ to OH- concentration that's equal. If I start going below 7, all the way down to 0 on the pH scale, my H+ concentration is increasing. Alright, so H+ is getting higher and higher and higher the further down I get closer to 0, which also means that my OH- concentration is decreasing. Go the other direction, get above 7, H+ is going down, but your OH- is going up. Once again, maybe not the worst thing to put on the scale in your notes. So 7 would be neutral, and your H+ plus OH- is equal to each other. Get below 7 all the way down to 0, and H+ is increasing every number that I go, but the OH- concentration is decreasing. Get above 7, every number I go, the H+ is getting lower, but the OH- is getting bigger. And something else to note about the pH scale in and of itself is the fact that every number on the pH scale is based on a log 10. So pH is a logarithmic scale. Why does that matter? Because that means every time I jump a full number on the pH scale, I'm changing the difference in H+ and OH- concentrations tenfold. Alright, so going from 6 to 7 is 10 times the difference in concentration in H+ and OH-. That's a huge jump. So if I'm causing some sort of change to happen by adding something to a solution and I'm changing it a full number or 2 numbers or 3 numbers on the pH scale, those are dramatic changes in those H plus and OH minus concentrations, right? If I go from 7 to 8, it's a 10-fold difference, but 7 to 9 is 100. 7 to 10 is 1,000 times different. So that really is a big change. So just keep that in the back of your head. So one of the things we've got to understand in terms of how acids and bases connect to living things is the fact that living systems don't deal well being outside of a particular pH range that they typically function in. Think of something even like your body and your blood. Your blood's got a certain pH, it needs to stay within a certain range, or you've got all sorts of issues that are going to come up with that. And so we've got natural buffers that exist in nature to help substances, help solutions resist large, drastic, dramatic changes in pH to keep things at homeostasis. So any buffer, whether it's natural or artificially created, works by accepting excess H plus ions when they're in solution, or if the opposite's happening, can also donate extra H plus to solution and thereby try and maintain a fairly consistent pH. Now, they're only going to work so well, but at least in terms of things like your body or our environment, it's better than not having a buffer system at all. So let's work through an example that does exist in your body. Alright? So natural human homeostatic pH should be somewhere between 7.35 and 7.45. Alright, not a particularly wide range, although when we remember pH is a log scale, it's not nothing. Okay, but it's certainly not jumping tenfold. We go anywhere below 6.8 or above 8.0, and you're dead. Alright, so in fact your blood is slightly basic. Okay, it's above 7. If we start going below that 7.35 mark, diagnostically, medically, they'll talk about that person's body being in acidosis. It's getting too acidic, right? It's getting extra H pluses in solution. That's not good for your body. Get below 7, and you end up dying as a result of your body not being able to maintain that pH. Opposite direction, above 7.45, we talk about your body being in alkalosis. Alkaline is another word for basic. So you're getting too much OH minus in solution or not enough H plus is hanging around. And once again, you get up in this range and you're also dead. So how does your body work to try and keep you here in that normal pH range? How do we keep you in homeostasis? Well, it actually gets connected to a bunch of molecules your body can produce, plus it also ends up being affected by your breathing rate. So when we look at your metabolism, and we start talking about cellular respiration later this year, one of the things that we're going to find is that CO2 gets produced by your cells. This is a natural byproduct of that cellular respiration process, your body's ability to take the food you eat and make energy so you can do things. And that CO2 is dissolved in your blood in the form of carbonic acid so that it can get to your lungs, and we can breathe it out again as CO2. Well, it's carbonic acid. So the more CO2 you're producing, because the more metabolic processes you're going through, the more energy your cells are demanding, say because you're out running that mile in gym class, that's going to cause that carbonic acid to build up in your body, making your blood slightly more acidic. So what ends up happening is that your body produces this bicarbonate molecule, gets produced by your kidneys, and that's going to cause that balancing back out. Alright, it's going to try and help bring your blood pH back into its normal range while you're demanding all of that extra energy. Okay? So, what happens, right? Your blood pH drops, which is where that extra carbonic acid's coming from because you're running that mile. Alright, so your body produces more of this bicarb solution and tries to remove that excess H plus from the blood. In addition to that, your body is also breathing heavier to try and get that CO2 out faster as well. Let's just say for whatever reason the reverse is happening. Maybe you're having a panic attack and you're not able to get rid of that CO2 as much. And so you overcompensate and your body now becomes more basic. All right, you're hanging on to too much of that bicarb. Your body needs to slow down that breathing rate, but it's also then going to kick up your metabolism a little bit, try to produce that extra CO2 and that carbonic acid, and try to raise your blood pH from there. So the idea here is that we want to keep this in balance, keep you at a consistent pH, so that everything in your body, all the molecules, work the way that they're supposed to.